Well, welcome everyone, and welcome to the 57th annual meeting of the Nantucket Conservation Foundation. I'm Arthur Gosnell, and I hope you will bear with me if, because of my reading glasses getting fogged up, I skip a step or two. Hopefully you'll catch me on that. But if I can, I will now turn to Mary Ann and check on our quorum and notice of meeting.
We do have a quorum present. We are okay to proceed. And I would like to ask to waive the reading of the minutes from the 2020 annual meeting. And then I would ask to approve the minutes of the 2020 annual meeting of members. So to vote for that.
So can we just have a quick vote on that, please? All in favor? Aye. Any opposed? Thank you very much. Thanks, Mary Ann. Well, terrific. Well, I'm going to make a few brief comments and then hand it over to Cormac, and hopefully I will not steal any of his thunder. As you all know, it's been a very challenging year for all 501c3 nonprofits, both here on Nantucket and across the United States. And I think many of these organizations are having their annual meeting right now, and many of those meetings are difficult because of things that had to be done and tough decisions that had to be made. Thankfully, in our case, because everybody just really did a spectacular job through this past year, whether it's staff members, board members, our supporters in general, we actually had an excellent year, and we didn't have to do any of the things that some of these other members had to do, such as furloughing employees and laying people off and stuff like that. So again, I don't want to steal Cormac's thunder, but I don't think our financial statements have ever looked this good, and certainly not in the last 10 years. In keeping with that, I need you all to remember that it was the support of our friends of this organization that mattered tremendously in this past year, and we need you to keep that support at a high level, and we need you to understand that we have more good ideas than we have money. So just because the financial statements look pretty good, we really need you to continue and to keep up your terrific support. But I again do have to just say thanks to everybody. The guys who took care of the properties, the land has looked wonderful all year, and we've had probably more people out on the properties over the last 12 months than at any time I can remember. Our science team did a great job, our financial team, our development team, our investment team, everybody. It was terrific, and we really appreciate all of your great work, and it mattered and it showed. And again, we're in a great place, whereas a lot of organizations are not. I also want to just mention that several weeks ago we lost a great friend of this organization. Jim Lentowski passed away, and he, as I suspect many of you know, was our executive director for 47 years. During that time span, Jim put together he structured hundreds of transactions to get land into conservation and to get our 9,000 acres assembled. And frankly, he went beyond that because he advised on pretty much any land conservation transaction that took place over the 47 years, whether we were in the lead or the land bank or whoever. So our thoughts and prayers go out to Jim's family, and we will miss him greatly. So with that, hopefully I didn't steal too much of your thunder, Cormac. I will turn it over to our president, Cormac Collier.
Thanks so much, Arthur. I'm going to pull it down just a little bit, if you guys don't mind. Before I do make my report, I would like to, on behalf of our treasurer, Charlie Gifford, who could not make it tonight, to give you a brief outline and report on our finances, if I can. Our fiscal year 2020 unaudited results are as follows. Total revenue for the period of January 1st through December 31st was $4,740,000. Of this, total contributed revenue was $3,554,000, making up that over 1.5 million in membership. And I wanted to just take a little break here and say that's the largest number in terms of membership that we've ever raised. And that's all in part to the people in this room. And we also are recording the meeting as well for our membership. So a very large thank you for that support. Over $400,000 for the Milestone Partners program, which has been a very successful development program where people have pledged $50,000 for over a period of 5 years. We had $114,000 in events, even though sometimes those events didn't actually take place, but those very generous people transferred that money to the support of the foundation. Approximately $290,000 in contributions to our land fund, $563 in permanent and temporary restricted donations, over $650,000 in grants, and $10,000 in estate and planned giving. Total expenses for that same period were approximately $3,643,000, bringing a net income of $1,996,000 approximately. I just want to state that some of that net income is as a result of permanently restricted projects that we will be working on in the future, but it alludes to the success that we had in the past year of both the membership stepping up and then the foundation doing its best during a very difficult period to put the necessary cost controls in place so that we really achieved a very strong and robust financial outcome. On behalf of the Treasurer, the Finance Committee, and the Board of Trustees, I am pleased to present this report stating a healthy and robust financial position as of year-end 2020 and continued momentum into 2021. The audited financial results will be completed by our auditor and reviewed by our audit committee in the coming weeks and presented in our annual report, which will be sent to the membership in the coming month. With that, Treasurer's report, done. Into my President's report briefly, because we don't want to listen to me. We want to see the keynote speaker, right? But let me give a brief update on where we went in the past year. As I said before during the Treasurer's report, first and foremost, a deep and heartfelt thank you to the membership and the Board of Trustees for their support, logistical, but also financial as well. It's the backbone of our organization, and we truly could not do what we do without that support. In keeping with the annual tradition of recognizing certain employees that have certain time milestones, I just wanted to point out that Richard Mack in our land management department has been with us for 15 years now. This is a funny one. Nick Larrabee has been with us for 10 years, and Nick has a head start on everybody. He started working in the cranberry bogs when he was a teenager. He's now the cranberry bog manager in his mid-20s, and hopefully he'll be working with us for many, many years to come. But he's already got 8 years on me. He's in his 20s. As Arthur did say, we did lose a legend. And I just want to echo his comments of thank you and appreciation for what Jim did to the organization, did to the land, and did for the community of Nantucket. There are very few individuals in the history of Nantucket that have such a long-lasting legacy as Jim has on the community. And it will be felt for generations to come, long after we're gone. And people are going to be enjoying the property that he helped protect. So, again, a thank you to Jim and all that he did. I want to add a few more moments of recognition to the entire staff. As Arthur had stated, we really rose to the occasion in the past year, it was a very difficult and somewhat trying time mentally for many people, but the organization never skipped a beat. And that's in large part, if not all part, to the staff that works here. Our properties, as Arthur alluded to and said, heaviest used that they've ever been used. And they were still beautiful. The land management department, clean, accessible, open for all. They put their heart and soul into it over this past year. And I just want to thank our land management team, Chris Iller, Don Mack, Richard Mack, Jake Visco, Bill Coffin, Jim Olney, and Brett Chetrell, our new ranger out at Cotu. And also thanks to Nick, as I stated before, for bringing in those cranberries during the fall season. Our education and educational programming and outreach was simply the best it has ever been. Our work grew exponentially from our core Mornings for Members programming to educational outings with our science staff that really provided a unique field-based insight into all of their work. Special thanks to Neil Foley and Alan Reinhardt. Neil, Alan, right there. Thanks so much. And also thanks to Grace Hull and Mara Weiner for all the work that they've done with marketing, events, and development, and the educational programming oversight. Thanks so much. As previously discussed, our financial position is solid and robust with the assistance and oversight of our recent 2020 edition of Tracy Finnerty as our Director of Finance Administration. She could not be with us tonight, but with a keen eye towards cost controls and near and long-term budgeting, we continue to show an excellent financial status from the end of 2021 and into today, 2020 and into today, 2021. So thanks to Tracy and Corylyn Adams for all their work in the finance department. Corylyn is here with us. Thank you. Finally, the science staff continue to work across all major disciplines. If it's out there, they're doing it. Botanical investigation, salt marsh and freshwater wetland restoration, rare and endangered species monitoring, nesting shorebird protection, and reviewing ecological responses to various land management strategies. Many thanks to Karen Beatty, Danielle Odell, Libby Buck, Kelly Omond, Jennifer Carberg, and this year's seasonal assistants, Chase Matthew and Nick Dunham. Thanks so much. So with that, I'd like to conclude my brief report and offer up a final additional word of gratitude. And that's not just to the members, the trustees, and the staff, but it's to the land itself. That's why we're all here. So thanks to the rolling moors, the sandplain grasslands, the beaches, the dunes, because without you, this past year would have been so much more difficult. So thanks. With that, let's get on with it. I would like to introduce Danielle Odell, our ecologist and field supervisor, to introduce our keynote speaker. I think we have a process, though. Arthur, if I could, adjournment of the business meeting. Adjourn business meeting. Danielle.
Hi, everybody. I'm Danielle Odell. I'm an ecologist at the Conservation Foundation. I am so happy to introduce you to Sam Hoff, who's going to speak to us today about her research. Since 2015, when we started learning about our northern long-eared bat populations on island, it has been a fascinating few years of meeting and collaborating with other bat researchers in the Northeast. I consider myself so lucky to have had the good fortune of working with some incredible women, including Zara Dowling from UMass Amherst, Liz Olson and Luann Johnson from Biodiversity Works on Martha's Vineyard, and most recently with Sam. All of these women have been incredible mentors and extremely generous in sharing their time and batting knowledge with me. All of us in the Science and Stewardship Department are very grateful to have had these fabulous research partners as we learn more about our northern long-eared bat populations here. I am so thrilled that Sam is here to talk to us about her work on northern long-eared bats in the coastal region of the Northeast. Sam is a PhD candidate at the University of Albany, and her interest in both disease ecology and bats has led her to focus her research on white nose syndrome, the fungal disease that's been decimating bat populations across the US. This led to a collaboration with the Nantucket Conservation Foundation, where together we have unveiled the importance of Nantucket in supporting some of the only remaining populations of the federally threatened northern long-eared bats in the Northeast. So without further ado, I would like to welcome Sam to talk to us about her research.
That down a bit. All right, thank you so much, Danielle. I'm really excited to be here on the island again and have the opportunity to speak with you all about the research we've been working on for the past 5 years. As Danielle mentioned, not just here on Nantucket, but we've been focusing on populations of these bats across the Northeast, particularly these coastal islands that we've found have remnant populations surviving in the face of a really devastating disease. So in case you're not too familiar with bat diversity in the Northeast, I know we probably have a lot of birders in here, but we'll be focusing on our only flying mammals that we have. I just wanted to give you a little overview of some of the species we find throughout the whole region. So the three that are all the way over here on the left, we have red bats, hoary bats, silver-haired bats. These are what's called tree bats, and they're migratory species. So they have been found on Nantucket mostly in the summer, and Nantucket also serves as a really important stopover during their migration further south in the fall. These other 6 species are hibernating bats or cave bats. So they remain in the Northeast throughout the winter, hibernating in caves and mines. The tricolored bat, little brown, Indiana, small-footed, and big brown, all found throughout the Northeast, and northern long-eared bat as well. However, the northern long-eared bat is really the only one that resides on Nantucket. There has been one record, as Danielle pointed out to me recently, of a big brown, but other than that, it's solely northern long-eared bats, which is really special. They are a very small bat, about 6 to 8 grams, so pretty tiny, and not many defining features. So when you look at the northern long-eared bat compared to these other cave bats, they look pretty similar. The only way to really tell them apart in the field is that they do actually have longer ears, and the little piece of skin that's in their ear, the tragus, is very, very long and pointed. So it can take a lot of work to become familiar with differentiating them. So northern long-eared bat is the star of our show today. And if you're not familiar with white nose syndrome, I just want to introduce a little bit about this disease. Really devastating invasive fungal disease that entered North America in around 2006, 2007, responsible for unprecedented mortality of 12 species at this point. So there's been 12 species that have been confirmed with symptoms of the disease, and a few others that have been found with fungal spores on them but aren't affected as of yet. It's named for this infection with the fungus essentially grows within the skin membranes, so face, ears, wings, and you get this sort of typical fuzzy white growth on all those skin membranes. And the problem is, sort of atypical for a fungus, is that this particular one likes to live in the cold. So it does very, very well in cave and mine environments where we find our hibernating bats. Most of the transmission at this point is from bat to bat when they are hibernating. Many of them like to roost in really large clusters. As we all know, we've had experience with COVID-19. Packing a lot of individuals in a small area can lead to a lot of transmission. So that happens with white nose as well, although it can persist in the environment in the caves. So spores can hang out indefinitely. As soon as a bat comes back in and picks one up, they can become infected again. And how it actually causes mortality, when it's growing within the skin membranes, it's causing periodic arousals. So bats, when they hibernate, they go through what's called torpor. They're not just asleep the whole time. And they typically wake up every few weeks, get a drink, go back into hibernation. The problem is when they're infected, it's essentially irritating. Of course, you have a fungus growing in your skin, causes a lot of dehydration, and it causes them to wake up more frequently than they normally would. So when that happens, they have to raise their metabolism, and they're burning up all that energy they saved for hibernation. So more and more arousals, more and more disruption causes mortality. If they do try to go out and fly during the winter, they're not going to find food to build back up that fat. And just the images on the bottom here, we have what the fungal spores look like. This one in particular is what it looks like under a microscope. So one of the characteristic ways to tell if a bat is infected is to look at the wing membrane under the microscope, which you can only do if they are dead. But you can see the little lesions growing within the skin. Another really quick tool to tell if a bat is infected is to use UV light, because all those lesions in the wing will actually fluoresce under UV light. So we can tell if they've been infected or not and how severely. And some of the common symptoms in the early years were abnormal behaviors, so bats flying out on the landscape in the middle of the day in the winter, and then a lot of degradation of the wing membranes, as well as any other exposed skin. So first arrives around 2006, 2007, right in the capital region of New York. There are a few caves nearby where some of the symptoms were first observed. Also just, mass mortality of a lot of these hibernating bat populations. Fast forward to 2012, 2013, you can see it quickly spread down throughout the southeast into the Midwest and up into Canada as well. And present day, we have reached the West Coast. We're now, the disease is now present in 37 states, 7 Canadian provinces, and a few others that are what we call suspect. So the fungus, PD, has been picked up, but the bats aren't experiencing symptoms just yet. The reason the map here looks a little patchy is that these are counties where the disease has been determined to be present and affecting bats, but that really just reflects where we find caves and mines throughout the US. So, if they were present in all the counties in all of these states where they have the disease, it would be completely covered. So bats do different things depending on the seasons. And the 3 main seasons I want to focus on that really relate to how this disease can actually affect these bats, so when it can affect them and where, are the hibernation season, maternity season, and then what we call fall swarm season. So fall swarm is a really interesting time. Bats are basically meeting up at hibernation sites. They're mating, but they're also foraging as much as possible to build up those fat reserves to sustain them throughout the winter. So this is when they typically will pick up the fungus. They're going in and out of sites, they're interacting with other bats, and they can become infected. As soon as hibernation takes place, this time period in the middle here on the graphs, many of these bats become infected very quickly, and it spreads throughout the whole population. If they are able to survive that winter, they can clear an infection, and they can make it out in the summer landscape, ideally to their maternity colonies where the females hang out with other females and raise their offspring. The problem is if they are so heavily affected by the disease during hibernation, they might not be in good body condition and they might not be able to reproduce that year. So even if we have survival throughout the winter season, we might still have population declines if they can't reproduce. And this is just showing some work done by one of my colleagues. And the main takeaway here is that different species are affected differently. So some, once they become infected, it reaches almost 100% of the hibernating population very quickly, while others, it spreads more slowly. And the fungal load, so the amount of infection they actually have, varies as well. And the highlighted bat here is the northern long-eared bat in the red-orange color. You can see they reach almost 100% prevalence in the population really quickly as soon as they enter hibernation, and they carry some of the highest fungal loads. All right, so why focus on northern long-eared bats? Well, they are one of the species most heavily affected by the disease. In the Northeast, we've had population declines anywhere from 90 to 99%. Many sites completely extirpated within 3 years after the fungus reaches that site. So the map in the middle shows the entire range of the northern long-eared bat, so it does stretch up into Western Canada. That's in the gray color, and then the red is the range that's been affected by white nose. So it's almost 80% of their entire range has been affected. Some other work done by two of my other colleagues, basically just showing the count of bats in hibernation sites pre and post white nose years declined drastically in what they call now, established years when the disease has been around within at least 5 to 10 years. And looking at the sites that remain, unfortunately, those where they are persisting, almost 90% of them only have less than 10 individuals left. So the populations that remain are very, very small. And again, really high fungal loads and prevalence within the population would indicate that they can't control growth of the fungus at all or tolerate an infection. However, enter our coastal populations, where we've been seeing some evidence to the contrary. And our research, as Danielle mentioned, focuses not just on Nantucket, but also on the east end of Long Island and Martha's Vineyard as well. Unfortunately, there was very little information about this species across all three of these islands in the pre-white nose years before it arrived to North America. So we really didn't have much to compare with, but what we have is presented here. So there was one study done of mammals on Long Island in the 70s. They did find that northern long-eared bats were the most prevalent species out on the east end, so particularly the forks. We also do have submissions to the rabies lab. So if a bat gets submitted for testing, it just shows that they occur in that area essentially. So we had pretty stable submissions from that data source as well. On the Vineyard, there was one master's thesis done in 1999. She did a lot of netting, so capturing a lot of bats. I think she captured 250-something bats, and 240 of them were northern long-eared bats. So it would seem like, they're the most prevalent bat on the island at that time. On Nantucket, the only study was really the work that Danielle and Zara Dowling from UMass Amherst did in 2015-2016. But what they found was a really robust population of northern long-eared bats. So they did acoustic studies, they did netting, tracking these bats, and even finding a local hibernation site, which you'll see is key to the success of these species, or the species. So what about in post-white nose years? The years that I have here are the year of first detection for each island. So that was 2011 for Long Island. Capture rates have declined, but they've pretty much kind of stabilized at a low level at this point. Same thing on the Vineyard. The first detection of the disease wasn't until 2017. Also seen a decline in captures, but kind of stable at a low level at this point. Nantucket here, first detection was 2017, but the first bat actually found with symptoms was just last year. And capture rates are still very, very high. So seemingly maybe something different going on each island. But through all of our efforts netting, we have documented successful reproduction still happening in the summer and multi-year survivors. So bats that we banned and we've captured 4 or 5 years later, even those that have been infected, which is really important. So the big question is really, what's special about these coastal regions that's supporting these remnant populations? When we think about, looking at some new invasive disease, these are the 3 things that you typically want to focus on. So, the disease itself, the agent, the pathogen here, PD, the host, or our bat species, and then the environment. And thinking about what's different on the islands versus the mainland. So why have populations crashed there and survived here? Could it be something different with the environment that reduces transmission? Is it something different about the bats themselves? Are they unique in some way? Or is it something about the pathogen? Maybe it's less successful here for whatever reason. So we set out to try to answer all of these questions as best of our ability. And the first thing we wanted to do, when you have a species that's threatened, federally threatened as this species is, and you don't have a lot of information about them, you want to basically know where are they and what's influencing that distribution first, so we can understand what we need to do to protect them as well. And to do this, one easy way is using acoustics. So bats fly around using their sonar to catch insects. They're constantly sending out these signals. And luckily, we have an awesome tool, bat detectors, basically, that we can go put out on the landscape. It's going to record all the ultrasonic sounds the bats make throughout the night. And we can go back in the office and spend hours and hours reviewing all those files and identifying them down to species and see where are we finding northerns, where do we not find them, and what's different about those places. So this is just showing you what those actual individual pulses look like on a computer for all the 9 species of the Northeast. So you can see they all have sort of a different little signature pulse that they emit. And to answer this question, we set up this grid system on each island. So 25 grids on Nantucket and the Vineyard and 50 on Long Island. We put 3 detectors each within these little grid cells and surveyed them during the summer when we typically have highest activity. And one thing to note, this bat is typically described as a forest interior bat. So they like really dense, cluttered forested areas, which, you know, there's not a ton of those on Nantucket, but you do have some. So we wanted to survey across all different canopy cover levels. Maybe they're using different types of habitat here than they would on the mainland. And that is really what we found. So I'm sorry this is really tiny, but I'll explain it. So all these little squares that are red are places where we found northern long-eared bats, and then the black is where we didn't. And then these are the rates. So essentially, if we put out a detector for one night, how likely are we going to be able to detect this bat? And what percentage of sites did we find them in? So Long Island, we found them in almost 60% of the sites we surveyed. On the Vineyard, 65%, and on Nantucket, 86%. So you'll see that if you can see, every little square on Nantucket is red because they were present in every grid cell we surveyed, just not at every detector location. But they're virtually everywhere, which is great news to us. And we were even surveying in some really open habitats, like this picture Danielle took here. And then we can look at, okay, what's different about these places where we find them and we don't? Is there any sort of environmental association we're finding? So Nantucket, not much really affected where we found them and when we didn't. This is looking at canopy cover or occupancy versus canopy cover. And as soon as the canopy cover is over 10%, it's like almost 100% likely they're gonna be there, which is great. On Long Island, really the most significant thing we found was development. So if you've ever visited Long Island, you know there's quite a difference in development on the west and the east end. And they were actually using areas of low levels of development where you might even have some fragmented habitats, but they're still using really tiny patches, even 1 acre or less, which is good news. But as soon as you get really high levels of development, like this, surrounding that site, it drops off and you're not likely to find them. So these are really, really useful information for us to use to not only understand what we can do to protect this bat, but to also just figure out new areas to survey. So next question, what are they doing in the fall and the winter? We know we have some evidence they're spending the winters here locally, but what does that look like? What local hibernation sites do we have? Can we characterize these sites? As you know from the geology of the islands, there's no natural cave system, so they must be using some other types of underground structures. So this is where all the fun fieldwork comes in. We get to MistNet to capture bats, and then we attach little tiny radio transmitters to the bats. So because they're a small bat, 6 to 8 grams, these transmitters are only about 0.5 gram at most, or 0.3 grams essentially. And that means that they don't last very long. So it's kind of tricky. We try to net as late into the fall as we possibly can so we can still catch them, but then also track them to where they're hibernating. And these are just some examples of places we have netted on Nantucket here. And then once you have attached your transmitter, you go out every single day and you try to find where that bat is. So what trees they're roosting in, and hopefully their eventual hibernation spot. So to do that, you have a radio receiver and an antenna. If you've ever seen Danielle driving around the island with an antenna out the window, she's not stealing your Wi-Fi, she's looking for missing bats. So it's a lot of fun. And over the past 4 years, we have tagged over 50 bats across the 3 islands. So a large number, which is really exciting. And just to give you an idea of what some of the roosts we're finding, most of these are tree roosts here on the left, but we also have a lot of anthropogenic structures that the bats are using. So about 40% of our roosts across all 3 islands were human structures, and on Nantucket, that was almost 60%. So I think it reflects what's actually out there in the landscape composition. You might think of a bat roost as a really large tree that has a lot of cracks they can work themselves in, and they will use some of these larger trees, but these bats are also not very picky. We found them in plenty tiny trees, just in a little dead limb. They do like little cracks and cavities if they can find one in a snag. With all the pitch pines here, Danielle is finding a lot of roosts just under loose bark or in a pitch pine snag that has a nice cavity. Even a dead limb hanging from a tree. They don't care as much. And then in terms of the human structures, a lot of times under the trim on the outside of houses, we found telephone poles, stove vents on the top of houses, and culverts. So they're taking advantage of what's out there on the landscape, which is a good sign. And what about hibernation spots? So we've found about 15 across the 3 islands so far, and the overwhelming majority are crawl spaces underneath houses. So that's a pretty common feature in many of the old construction styles across all 3 of these islands, and it really mimics a cave environment very well. It's damp, it's cold, it usually doesn't go below freezing, which is important, so they're safe throughout the whole winter season. And most of these houses have these sort of great entrances that the bats are using to get inside. And most people really had no idea that they had bats in their crawl spaces throughout the whole winter. We did find a few sites that were larger. So on Long Island, we actually had a old World War II bunker that the bats were using. And this was really the largest size site that we found, where we also had other species within it. But the really interesting thing is that in majority of these sites, only northern long-eared bats, so low diversity. We also have very small numbers, so low densities. No more than 5 bats did we ever actually see in a site. But this is important in terms of the disease dynamics. So low density is going to lead to less transmission, of course, from bat to bat if they pick up the fungus. And less diversity is important because some of the other bats are kind of super spreaders, so we don't want them around. It's a good thing. So again, I'll explain these graphs because I think it's probably going to be hard to see the axes. We also wanted to measure, what's activity look like during the fall? How are they using the landscape? Are they actually present on the landscape later than they would be in the mainland? So we put our detectors out in the fall as well, and this is from September through the end of December. Most bats on the mainland are going to start hibernating in mid-October, maybe late October. Here, we have these big spikes in November. So bats are still out on the landscape. We even find them still roosting in trees almost to the end of November, which means that it's got to have something to do with the weather. I think if you live here year-round, you probably have a more mild coastal climate than you do further inland. So this is allowing them to stay active later in the year. And then looking at, a whole hibernation season, bats are able to be active later in November. They can start becoming active early in March. So we have a shorter hibernation season overall. It's about 6 months on the mainland versus 4 months here. So if bats do become infected, less time for the disease to progress, and they can, presumably survive the winter. This didn't show up very well, but I wanted to see what weather variables might be influencing activity that we see throughout the winter. Really, the most important thing was the maximum daytime temperature. So if you guys are present on the island across, the winter season, you probably know that you have less days that go, into the freezing temperatures than you would on the mainland, and less long periods of freezing temperatures when you do have them. So there's more opportunities for bats to come out on the landscape. If it's above, 5 degrees Celsius, about 40 Fahrenheit, it's a 50% chance they're going to emerge that night. So we do actually see a lot of activity throughout the winter months, periodically on those warmer winter nights. Now you might think, white nose, more activity, that's a bad thing. So we wanted to answer that question. If they come out of the winter, is there anything for them to eat on the landscape? So these bats are insect specialists. And my colleague at Albany, Casey, who's here with us too, wanted to answer this question. So she facilitated a large volunteer-based survey where we actually had volunteers go out and do insect surveys throughout the winter. So we used light traps and sugar bait traps, and they went out on those nights that were 5 degrees or higher to see if we were getting any insects. And it turns out we got a lot of them. I think it was almost a 70% success rate if it was 5 degrees or higher that night. And of course, the warmer nights, a lot more insect activity. So this is insects up here, northern long-eared bats here. We were pairing acoustic detectors with our insect traps. And you can see it aligns pretty well. We're getting a lot of activity, obviously in the fall and the spring, but throughout the winter as well, of both bugs and bats. And the bugs were even coming out on cooler temperatures than you would expect them to be able to fly at. And if you have any bug-related questions, I'll give them over to Casey later. But in terms of species composition, northern long-eared bats are a moth specialist, but they will eat anything they can get in their mouths, really. That's the right size. So beetles, flies, even spiders, they will actually snatch insects off vegetation. So they don't just have to catch them flying through the air, which is really cool. They have two strategies. And some of the moths we were getting in our insect surveys, we had some moths that are called fall moths. So, they're usually only found in the fall, but they were found throughout the winter. We had some that only emerge in the spring typically, but they were, present earlier in the winter months. And even some migrants that actually come up from the south that were arriving earlier than you would expect. So seemingly a decent amount of food diversity out there for these bats. So we have all these factors kind of working together to support these populations. The last thing I wanted to show in terms of results is what does the disease look like? So every time we catch a bat, we take a swab sample, send it off for processing, and we can see if they have the fungus on them or not. So that's kind of what that looks like. Take a little swab, it gets processed. So Long Island is the green, the Vineyard is the purple, and then orange, Nantucket. And this is just, prevalence, essentially. So how many of those samples did we collect and how many were positive? Really low numbers in the fall, as you would expect, because that's just when they're kind of initially picking up the fungus. And then in the spring, about 70% of our samples were positive on Long Island, 100% on the Vineyard, and we've only ever had our one positive case on Nantucket so far that was confirmed. So it seems like something different is kind of happening across each island. Maybe they're in different stages of invasion, and I think it could have something to do with the spatial isolation of each island. So the Vineyard is the closest to the mainland. Depending where you measure on Long Island, it's kind of in the middle, and Nantucket is the furthest. So there's probably very little migration, if any, between Nantucket bats and mainland bats going back and forth. And of course, in the spring, we look at them with UV light as well, and I just wanted to give you an idea of what this looks like. So this is a little brown bat on the top from mainland New York, and that's like a really severe infection, what it would look like. We have the Vineyard here and Long Island, kind of worst case scenarios. So they're not even really very comparable to the worst case scenario on the mainland. So seemingly less of an infection. And more typically what we see is just a few little spots where they might have been infected, but they emerge in the spring pretty healthy and can make it onto the summer landscape and hopefully reproduce. So we just thought about this recently, but what else are we lacking on the islands? Well, lower species diversity of all those other hibernating bats, and little brown bats in particular are, essentially super spreaders at this point. They're kind of the largest reservoir of the fungus in the Northeast. So if you've ever heard of typhoid Mary, that's, call them typhoid little brown bats because they're kind of just spreading it all around to the other species. And just looking at our acoustic results over the past few years, it kind of aligns with what we were seeing in terms of the disease dynamics. So all other bats recorded are in blue, northern long-eared bats in green, and then the orange, which you can barely see across each island here, is little brown bats. But we do record the most little brown bat calls on the Vineyard, and much less on Nantucket and Long Island. So it seems like something's going on there, less migration between the mainland and the islands, which is good for our bats here. Okay, so to sum it up, we have all of these mechanisms supporting these populations and their ability to persist on our really unique coastal areas. We have, local hibernation site. That means, they don't have to migrate back to the mainland to hibernate, less energy and time spent doing so, less diversity in these sites, which is great for them, and they're not spreading it around as much, even if they become infected. We have opportunities for feeding throughout the winter and drinking as well. Open water sources remain as well on the islands. And, food available, again, is important. So this is great. It really speaks to the idea that these areas are refuges for this remnant species. They're not doing well anywhere else. So it's really important to protect what we have left here, and particularly in Nantucket, because it seems like if it is a spatial isolation thing, it's great because we can keep the numbers low in terms of white nose infected bats on the island. So what about current threats? We know the species is not doing well, even if they are doing better here. They're federally threatened, but actually they've been proposed for an upgrading to federally endangered just because populations are crashing throughout the rest of their range. So what other threats have we identified? Typically, summer habitat is one that people think of, trees. But you guys have a lot of protected habitat on the island, and the same on the Vineyard as well, and even on the east end of Long Island. Trees are not necessarily a limiting factor, and we know they're using small patches of habitat. I mean, clearing a large patch of trees on this island would not be good, but again, you have a lot of protected habitat, which is great. But what about winter habitat? So that seems to be the main limiting factor for these populations. Crawl spaces, they're out right now. If people are building new houses, they're not going to put a crawl space in it. They might opt for a full basement that they seal off or no underground structure at all. I'm sure you've seen a lot of older houses being renovated. They might choose to seal up their crawl spaces or exclude them and unknowingly harm hibernating bats. So that could be a potential limiting factor for all of our across all of our populations here. And disturbance is a big one. So, many of the houses we were finding were seasonal homes. Again, there's not going to be a lot of human disturbance. But some of ours are heavily disturbed, like our bunker on Long Island. People like to break in there year-round because they think it's, some site of a conspiracy theory. So again, disturbance in the winter is not good. It's going to cause more arousals and potentially harm these bats that are staying here. So our proposed solution is to build some artificial structures that these bats can hibernate in. And this is something we've been thinking about for a few years, but our whole project team just applied for a grant to hopefully be able to test this method. So it would be something very simple to build, relatively cost-effective, low cost in terms of, you're basically just building a crawl space and covering it up with some plywood and dirt instead of a whole house. It would have a similar entrance because they're used to using these little grate-like structures, only be 3 to 4 feet deep, walls of cinder blocks and plywood top. So we know we can build them. We've been monitoring the microclimate of all the sites the bats are using. We know what conditions they like. The question is just if we can get the bats to use them. So we're proposing putting them in areas where we already find the bats, already occupied habitat essentially in the landscapes that they're using in the fall already, and testing out some different methods to attract them to these structures. And bats are pretty curious by nature, so we think if we can build it, they're gonna come and check it out and hopefully use it throughout the winter. And the other important feature of these sites would be that we could decontaminate them if they do become infected with the fungus. So there's been a lot of research in terms of ways to decontaminate, sites and gear and things like that. And a really promising one is antifungal UV light that's already being used in hospitals. But you could essentially cover one of these sites very quickly and get in all the nooks and crannies and decontaminate the site if we find bats are using it, just to make sure we're not further spreading the disease. All right, so big thank you to NCF for hosting me. I love the opportunity to come to Nantucket. My first visit was last year, and I'll come back anytime. Thanks to our whole project team, a lot of people involved, as Danielle mentioned, NCF, Biodiversity Works on the Vineyard, New York State DEC on Long Island, many other people in our labs that have been involved in this work, and of course, all the landowners. We have so many great landowners that let us put up acoustic detectors on their property or crawl around in their crawl spaces throughout the winter and look for bats, which has been awesome, and people just reporting their bat sightings, because that has led us to a lot of sites as well. And of course, our funding, we've had some great luck getting some grants to do this work, and hopefully more in the future. So I think that's it. Thank you, guys. Yeah, I'll welcome any questions, thoughts, concerns. The what? Oh, good question. Yeah. Europe or Asia. As of right now, the closest genetic sample that's been taken, I think, was in Western Europe, maybe France. So it's not entirely clear yet. The fungus actually has more diversity. Everywhere they've looked in Europe and Asia, they've basically found it in all the caves and mines there, but it's not affecting their bat populations. So likely it's been around for a long time, and the bats that remain now have co-evolved to tolerate it. Yeah. Hopefully, yeah, that would be great. It's just some are declining so quickly that it might not happen quick enough, of course. But yeah, especially because we do see differences between the species here in North America as to how affected they are. You know, some just seem to be able to tolerate it very well and others can't. So there's been a ton of research from other labs looking into what those differences are. Is it some immune function? Is it, you know, the microbiome that's naturally present on their wings? Is it just where they choose to hibernate in a big site? Maybe they like very, very cold areas where the fungus can't grow. So it could be a combination of things for other species.
What are some of the larger implications of the declining bat population throughout the United States and worldwide, biologically, environmentally, et cetera?
Yeah, good question. Yes, he said, what are the major implications for bat declines, I guess through North America, but maybe globally too, yeah. I mean, I think bats are an awesome species, so just because they're unique, but also because they're very diverse. So there's over 1,400 different species of bats. They play many different roles. So ours are insect eaters. They like to eat a lot of insect pests, so all the things that bother us that are flying around at night, but also a lot of crop pests that can damage our agriculture and forestry industries. You have other bats throughout the Southwest and more tropical areas that are pollinators, so a lot of the foods that we like to eat and other things we like to drink are pollinated by bats, so a lot of importance there, I think. And yeah, I just think supporting, you know, a large amount of species diversity is always important, but of course bats are unique as well. You know, they're our only flying mammal, so yeah, important to support their populations. Anything else? Yes. It's hard to get population numbers with bats. So the acoustics, although it can tell us where they are, we can't get, you know, numbers of individuals because, you know, it's a stationary detector. One bat might fly back and forth all night versus 100 bats flying around. But something to look at that would be important is like activity levels to see if that's declining over time. And that seems to be remaining stable. You can try to infer from that if you look at capture records year after year, and that's pretty much remaining stable, if not even maybe increasing a little bit on Nantucket. So all signs are good so far. Yeah.
Thank you all for coming, and we look forward to seeing you next year. Thank you.